Okay, so I will go over into the lab, but I think we should start with some requirements for sample collection first, before we start testing. Okay, so I will talk about a little bit of the methods, but not in a lot of detail, just more to explain some of the considerations. And as Prof Markotter mentioned, we can share the SABRINet manual that we have put together with more detailed methods and things to use when embarking on a biosurveillance study. Okay, so my talk will just be structured in 3 different sections, which would be in-field sample collection, looking at both from the environment and then from individual bats, over to sample testing, which would be nucleic acid and serological, and then lastly looking a bit at virus characterization, which can be just basic identification of what you've detected, and then also some additional characterization that can be done. Okay, so I would just like to start off, and as you might recall from Dr. Epstein's talk yesterday about different study designs, so just to mention again, cross-sectional studies is where when data is collected at a given time point in a specific population, and these can also be opportunistically collected samples, which means the study is more unstructured compared to a longitudinal study. With studies like these, you will be able to report on the diversity of viruses present and maybe also identify some new host species in which these viruses can be found. Then longitudinal studies, which is more repeated examination of specific individuals or populations, where you can detect changes that might occur over a period of time. So as you saw in the previous talk, with mark recapture methods, we are able to look at specific individuals that we sample multiple times depending on when we actually recatch them. So these studies would then be more structured. It can also provide information on the diversity. You can also provide information on the excretion of these viruses into the environment and how the dynamics change over time. We can look at changes in infection status from individual bats, or changes in serological status from individual bats. And then these studies can be designed to collect samples either monthly, seasonally. Some studies also report going weekly. But as you can imagine, that this is labor intensive and can take a lot of field time. And as we had the working group discussion yesterday, some people pointed out they don't have a lot of storage. So that's also a consideration when you do longitudinal studies, that you make sure that you have the space to store these samples when working on it. So this will also, the study design will also have different resolution of your data, where longitudinal studies will give you better resolution on your data. But what should also be considered is the type of samples, and that would be collecting from individuals compared to collecting from the environment, or at population level. So what do we mean when we talk about collecting from the environment or at population level? So that would be collecting of bat excreta like urine and fecal. So this would be usually from underneath the roosting bats. That is why we call it from the environment and not the specific individuals. So this has become a very popular approach to use since it's not invasive. And there's limited disturbance of the bats. At most, it would be roost disturbance. For instance, when you enter into caves, the bats would tend to fly around a bit. And I think the ethical considerations when conducting such a study is not as stringent as it would be when you're actively capturing and sampling from individual bats. So before I go into the methods that we use, there's a few considerations that I think you should take into consideration when designing such studies. So the first would be the time of sampling. So when collecting environmental samples, obviously you want to collect them when the bats are in the roosts, which would be during the day, as compared to individual sampling when we sample at night, when the bats move out to go forage and they fly around. What we found is that we like to go collect the samples, especially from our cave sampling site, early in the mornings, not very early, but earlier in the day, after the bats have returned from foraging, so we can collect fresh urine and fecal droppings from the environment. Then also what I would like to refer to as active and delayed sampling. So we would go, place sheets underneath the roosts, and actively collect the samples for maybe 2 to 3 hours until we've reached the numbers that we want to collect. Versus some studies report on what I refer to as delayed sampling, where they would put the sheets, leave it for a few hours, come back and collect the samples. So with the latter, you have the problem of, let's say, you put it underneath an aggregation of a few bats, and there might be 1 or 2 individuals that are actively shedding virus, and as they move around or they defecate or urinate throughout the day, they can perhaps do that in multiple places on your sheets. So when you're collecting samples, there might be an overestimation of the prevalence of this virus, and that should just be considered when doing data analysis. So the next consideration, So the next consideration would be the roost type. So as Lo mentioned in the previous presentation, bats don't just roost in caves. We have different species that would roost in trees, roost in the roofs of houses. Examples would include, especially for sub-Saharan Africa, is Eidolon halvum, that roosts in trees, Rosetus aegyptiacus, that would roost in caves, and then you get a lot of insectivorous bats that tend to roost in the roofs of houses or abandoned buildings. I just included a picture here at the top, which is a few Epimetheus from KwaZulu-Natal that would roost in the roofs of thatched houses. So considerations there would be for caves, it would be accessibility and safety. I mean, if you enter into a cave, you would need head protection and for your own safety. And is the cave actually accessible without the requirement of ropes, or is a flat surface, can you move in? And then also, is there spaces to actually put your sheets underneath the bats that roost, or is it irregular surfaces where you won't be able to put your sheets for collection? And then in terms of houses, for instance, the shape of this house here in the corner is typically what we have, and where the ceilings are slanted, so the bats can roost in the corners, and it's not always accessible to humans to reach those areas to place your sheets there. While for houses where there's no ceilings installed or thatched roofs, as you see here with the Epimetheus, it's a bit more easy to put your sheets down to collect the samples. So the second consideration would be the roost community structure. So if we collect samples from an environment, we can't with 100% confidence say that this sample came from a Rosetus, for instance, because we didn't actually collect it from the individual. But if you know a bit more about the roost community structure, you can with a high certainty say that the samples that you collected are in fact from the species. So we do know that certain bat species tend to co-roost, let's say for instance in a cave. And then we also have species that would aggregate together within the specific roost, as indicated here by the image, which is Myotis and Rhinolophus that would aggregate together. So when you collect your samples, it's important to consider this, because if you now put sheets underneath a mixed aggregation of species, you won't be able to, with high confidence, say, okay, so the virus that I detected in the excreta is specifically from a Rhinolophus species, and you would only be able to say, okay, it's from insectivorous bats within this cave. So just an example of the cave that we do, where we sample, the Matlapitsi cave, we know that Rosetus tend to roost within this large open big cavern in the cave. So when we collect samples, we can with relative certainty say that the samples that we are collecting are in fact from the species, as we know that the insectivorous bats roost more within the outside smaller caverns within the caves. Okay, another consideration would be biosafety. I'm only going to touch on this a little bit, as Prof Markotter will go into a bit more detail regarding biosafety and PPE and things like that when conducting the research. So when we enter caves, we tend to use full PPE for our safety and for the bats, actually. So there's just, the image is labeled with a few key things that we have, like head protection, respiratory protection, and then boots when we enter into the caves. There are alternative options where you can use hard hats, N95 face masks, and goggles. And what's also important is decontamination. If you're underneath a roost, whether you climb into a roof, whether you go into a cave, you do have the bats that would defecate and urinate on top of you. So it's very important to do proper decontamination when you come out of the cave. And you also want to not introduce pathogens from one cave into another cave. So that's why it's also important to consider decontamination of, for instance, shoes and things that you regularly would use at the different sampling sites. Okay, so let's get into some of the requirements for doing sample collection. So the first would be sheets or trays. So it is important to make sure that the sheets and trays that you use are sterilized and pre-treated with things like DNAs and RNAs, which specifically we use to prevent any contamination of enzymes that might degrade the RNA or the DNA of the samples that you collect and affect downstream processing of your samples. Then another thing would be, should these sheets or trays be suspended up into the air, or can you put it on the ground? So we do put it on the ground because we do active sampling, so we are constantly there to see if there's anything that's running over it. But a lot of studies prefer to suspend these sheets within the air, especially if you leave it for longer periods, to prevent any other animals like rodents or things running over it and contaminating the sheets or things like that. Then between plastic trays and sheets, we prefer to use the sheets because they're more rigid. So it gives you a flat surface. And as you can see in this image where they suspended some of the sheets, you do have pockets that form within the sheets. And you can have a problem where some of your urine and things can run down and pool in pockets. And then if you do pooling, it complicates the situation. So we prefer the plastic sheets that are more rigid to be able to collect the individual samples as we go on. Okay, so another requirement is what you actually use for collection of the samples, which would be swabs, forceps, and or pipettes. Okay, so for fruit bats, and for our research, we specifically work with rosettas, so it's quite a small fruit bat compared to the larger species, like some of the work that they've done on Nipah virus, as previously talked about, with the larger fruit bats. So rosettas doesn't urinate as much as you would see for the larger fruit bats, where it's milliliters compared to microliters. So we tend to use pipettes to collect the urine droplets, or alternatively swabs. For the insectivorous bats, for urine collection, you can use pipettes, but we also tend to use the swabs because it's such small volumes of urine, and if you use pipettes, you tend to sometimes lose some of this volume, and it works a lot better for us. In terms of fecal collection, so you can actually distinguish between the fecal of a fruit bat compared to insectivorous bat. The fruit bat's fecal matter is a bit more pulp-like, while you have pellets for the insectivorous bats. So to try and pick up fecal matter that are more pulp-like with a tweezer is not going to work, or not going to work as well. So we use swabs to collect those as well. But for the insectivorous bats, you can use either. The problem with tweezers or forceps is that you will have to use clean pairs between the samples that you are collecting, otherwise you're cross-contaminating your different pools. And with the swabs, it works quite nice if you collect, for instance, 3 fecal boluses or 3 urine droplets. You can put the swab within the tube and just break off the end, and just there's your sample, and you can continue. So I just highlighted here in blue. So sample inactivation, what we do is we would pre-aliquot the inactivation media or the transport media, which in this case we use DNA and RNA shield, into the tubes, which makes it easier to just put your sample in there, you close the tube, and it's ready to ship to the lab. Another consideration is a lot of studies that do environmental or population level sampling make use of pooling, which allows you to actually collect more samples. And when pooling it, it reduces the cost, and you can actually, especially if looking at diversity, you can include more samples, if I can call it that. However, pooling can lead to an overestimation of prevalence, and this paper by Giles et al. Had a nice image explaining that depending on how you pool, you can see that there's, if it's the same population, you can end up having different prevalences of the viruses within the samples depending on your pooling strategy. And there are ways to optimize this, as here where they did quadrant pooling, to get your number closer to what you would actually see in the individual population if you would have looked at that. So this is also very important when conducting your data analysis after you've collected these samples. Okay, so to move over for the collection of samples from individual bats. As you've seen in the previous talks, there are various ways in which we would capture these bats before we start sampling. So then we have the catch and release sampling, and then voucher collection, where animals are sacrificed to be able to get different samples. So for catch and release sampling, we collect a basic set of samples, which include blood, which we get serum from, and then we take wing biopsies, which can be used for molecular barcoding to identify the host species, oral swabs and rectal swabs, and then more opportunistically, we would collect urine and fecal, as it's not always as easy as seen in the previous or in yesterday's presentation, where they have the tubes at the bottom of the bags to collect the urine. Because the smaller bats and rosettas, actually, if they urinate, it's not a lot, so it gets absorbed into the bag, so you lose it. So we rely on in hand, if the bat urinates, we collect it, or fecal matter in the bag. So I've just indicated a few more sample types that other studies have collected from catch and release sampling in blue. And then to move on to voucher collection. So if we do take vouchers, we would take all the visceral organs, including the tongue and the brain, and then again some other samples in blue, which other studies have also collected depending on your research question. So we feel that if vouchers are collected, even though you're only testing for a specific virus, let's say your study looks at coronaviruses and you want to look at the intestine or the rectum, collect all the other samples and keep it. Use the resources that you have. Keep it on the side. There might be future projects and things that these can be used for. Alternatively, these samples can also be used to look at the tissue distribution of these viruses throughout the bats. And in the event where wing biopsies is not successful with doing barcoding of the species, you can use some of these organs, example heart and kidney and things like that that we have used in the past for barcoding, and it has worked very well. Okay, so this is, I just included this to show you a general setup of how it will look when we go out into the field to do our sampling. So on the left here, we would have the tattoo station, as Lau explained in the previous presentation, and how we do that and everything that we need for that. And next to it would be our sampling station. So with here, I just want to show that there's additional small things that you need to consider, even though I'm not going to go into detail for that, and things that you will need, like your bio-waste, how you would handle that. We would have sharps containers for things like your needles or your pipette tips, and then just bio-waste bags for other waste that you generate. It's also important to have a decontaminant on hand, because you never know if there might be blood that spills onto the surface where you're working, or the bat urinates, and you want to clean that up before you continue to not cross-contaminate the samples or your working surface that you're working with. And then general things like pre-labeled tubes and sample racks and everything. And if you have a layout that is set up, it helps you to streamline the process and make sure that there's limited issues coming in or personal injuries that happen if you're prepared properly. Okay, so like I said, not a lot of detail on the methods, just going to highlight some things. So with wing biopsy, it's for molecular collection, and I think the important thing to consider there is the size of your punch. So what we used is disposable biopsy punches and a wooden board, where we would stretch the wing out on and then take the punch from the wing. So you do get different sizes of disposable biopsy punch, but you don't want to put a 5mm hole in a bat that's like 4 grams. So it's very important to look at the target species that you're working with and plan according to that what size biopsy punch you would need. So just in general, we would take the punch, and punch both wings as close to the body as possible, but avoiding any veins that you see in the wing. There are some studies, you can go read up about it, where they look at the recovery rate or the healing process in the wing, and it's quite safe for the bats. So then to move on to swab collection, oral and rectal. Oral sample collection is quite straightforward. You would use a swab the inside of the bat's mouth, the cheeks, the tongue, to collect some saliva. The rectal swabs is a bit more complicated. As you can imagine, that cavity can be quite small for the smaller bats. And we've been able to get rectal swabs from smaller bats using some of these smaller tipped swabs, ranging from 1.5 to about 2 millimeters. There are smaller ones available that go down to 0.5 millimeters that might be more successful on the really small species, but we are actually struggling to get a hold of those. When taking rectal swabs, we do put the swabs into PBS, which is, it's like saline water, just to make it wet, to make it easier for the swab to go in as to not hurt the bats. But for the smaller species where we actually can't get rectal swabs, as I mentioned, we can get opportunistic fecal and urine collection during the handling of the bats. So it's important to just check the bag. As you take the bat out, there might be some fecal deposits in there that you can actually take as a fecal sample in the event where you don't have a rectal swab. In terms of the urine, and I think this is largely dependent on how much you work with the species, you can get to know it. Rosettus doesn't really like to urinate in hand, so we're actually struggling to get urine samples from these individual bats. But what we have seen over the years while working with Miniopterus, if you take them out of the bag and you open it quickly, then they urinate, and we have actually had really good success with getting urine samples from almost all of the Miniopterus bats that we actually take out. But this you will learn as you start working with the bats and the different species. For blood collection, so we do that to collect serum, and serum is then used in serological studies to look at antibodies of different pathogens that we want to study. So things that you will need for blood collection are needles to make the little hole where we can collect the blood from, serum separator tubes to split your serum from the blood, then pipettes and pipette tips, but you can also use glass capillary tubes. We don't use glass capillary tubes personally in the field because it is glass. You have the chance that it might break and it could result in injury. So we also prefer to use pipettes because it is quicker to get the required volume of blood that you have to collect, and then cover the wound to stop excessive bleeding much faster. So the process would just involve, you would collect the blood, you would centrifuge the tubes to be able to separate your serum from the blood. You would heat and activate it because we do not put these samples in an inactivation media. And then you would aliquot your serum into a different tube to use. So equipment that you would need for this, and we usually take this into the field to have everything done and inactivated there, would be a centrifuge to spin your blood down and a heating block to inactivate. So considerations for doing this would be the selection of the bleeding site. So there are a few studies that talk about where on the bats can you, from which veins you can collect the blood. So you have in the wing, the vein there at the cephalic vein there at the end of the wing. It can be the vein in the tail or the vein located on the humerus. What we've seen is, for some species it works well in the tail, but for the smaller species, sometimes the vein is too small, and then we move to the wing. And we would use the vein by the humerus as a last resort, because they tend to form hematomas if it's not done properly. What we've also found is that you struggle, especially with the small bats, to keep the tail open, and with multiple people handling the bat to be able to make the little hole to collect the blood, and the bats tend to move. The wing is actually much easier to control the bat, hold it down, and to get the blood from. So obviously your needle size is very important, and is also dependent on the size of your bat. We currently use 23 gauge needles for the fruit bats, and the very small 30 gauge needles for the insectivorous bats. Then another consideration would be the volume of the blood that you collect. So we all of you received a nice book as part of the workshop package. And in there you will see that there's a little table for each of the species indicating the average weight, the minimum weight, and the maximum weight of males and females. There was a study in 2010 by Smith et al. Where they looked at the collection of small volumes of blood from insectivorous bats. And in the end, their conclusion was that it's safe to collect 4 microliters of blood per gram of the bat's weight. So I just made a little table based on some of the South African species that we frequently work on, indicating the volumes that you would typically like to collect from the bats. And as you can see, it can go up as high as 660 microliters for a large male rosettus, as compared to 25 microliters of the smaller bat, like for Rhinolophus simulator. What's also important is trying to separate the serum from these very small amounts of blood is not easy. So we tend to dilute the serum of the insectivorous bats in field using PBS, and we do a 1:10 dilution in the collection tube before we spin it down. So this must just be kept in mind when doing subsequent dilutions for your serological assays, that you've already done a 1:10 dilution of the specific sample. Then moving over to necropsies where voucher specimens are taken. So I put in the sentence here that we discourage the collection of voucher specimens as far as possible. And I'm not referring here to the voucher specimens that we collect for new species or building up things regarding to taxonomy and things like that. With this, I mean going out to the field, killing 20 bats just to be able to get samples for virological research, especially if we can get away with using catch and release methods and getting samples from that. But there are, during routine sampling, you will have instances where you could have accidental deaths of bats, or some bats that are injured that require to be euthanized. It's important that these bats are also submitted to museums or collections and not just thrown away after samples have been collected. So to euthanize the bats, we would use an overdose of an inhalation anesthetic, for example isoflurane, and then do cardiac exsanguination where we remove the blood through the heart. So just general things that you would need for a necropsy would be scissors, forceps, and I included here swabs and needles. So when we submit the carcasses for museum collection, you would only want to make a little small incision in the skull to collect the brain, and obviously you won't be able to do that using scissors and forceps. So we then use the swabs to go into that little hole and collect the brain like that, to keep the skull intact as possible. And then the needle, sometimes during necropsy, you would see that the individual's bladder is full, and since we struggle to get urine samples from a lot of bats, this is nice to use the needle to just extract the urine from the bladder before the bladder is removed during the necropsy. Considerations would be biosafety, because you're working with the organs of the bat, there can be blood everywhere, so us, for instance, we would use full PPE and respiratory protection when we do necropsies of the bats. And also in terms of preserving the samples, we would split the samples upon collection into 2 different tubes, where we would have a stock tube and a working tube. The one we will use for extraction, the other one we would put away and use that should we want to go back to that particular sample. Before I continue, with our discussion yesterday, we talked about why it's important to know what research has been done in your country, even if it's not disease-related, even if it's just bat-related research. And I think it's important to also utilize historical samples. Some other research groups that might done work on bats might have collected samples to look at dietary analysis or something, and there might be fecal samples that can actually be used to supplement your research, and you don't always have to go and collect a lot of samples if you have samples that you can start with, and maybe build collaborations with these people and then maximize the use of the samples that have been collected. So just quickly on data sheets and sample storage. So it is very important to keep a good record of the samples you collected, because without that, the samples are actually useless if you can't link it to an individual or a location or a sample type. So what we do is we have these sheets, and I only put up a few examples for you, which is a sample sheet, an necropsy sheet, and then our environmental collections sheet, where we log different things. These are also in the back of the manual, where we provide all the SOPs for what we do. So we would complete this in the field, and then we would scan this to make sure that we have electronic copies if anything should happen to the originals. And we would also enter the data into a cumulative database. And you can either use Excel, or there's a lot of online programs that are available. As Kendra pointed out yesterday, she also mentioned Airtable, which can be used. And if I recall correctly, I think it's free up until the larger databases up to 55,000 records. Okay, so, but I do know, I do know for the smaller databases you can use it for free, but as soon as you go over, I think 55,000 records, you will have to then pay for a subscription. And then it's also important to, in the databases where you log this information, is to keep an inventory list of what you have available. Not only is this important for you to know for future studies what you actually have, it's also important for collaborations, and for us it's important for audits to see what we're actually doing with the samples. Do we keep record and what's happening around there? So then long-term storage, samples should be stored in a -80. And we also spoke about this yesterday, where people say they don't have access to massive storage facilities, and they are limited to storing in single freezers that they share with other people in the department. But it's just important to store sample material, especially if you want to keep it for a longer term, and then your extracted RNA and DNA at -80. Okay, so moving on to the actual lab work. So nucleic acid testing, I'll start off with that. And let's look at assay selection. So the considerations here would be, what is the actual aim of your study? And will you be able to compare what you've got with studies that have previously reported things? So I've just put here targeted surveillance studies. So certain studies will particularly look at a specific virus or a specific genus of interest, compared to more other biosurveillance studies that will actually want to look at a larger diversity and not just a specific virus. So some of, if you move up and you're trying to look for a larger diversity, you will be able to detect some unknown things, and a lot more, you have the probability of detecting a lot more viruses compared to using specific assays. But if you use this, you will then lose some of the sensitivity and specificity that you would have when targeting a specific virus. Just for instance, this is just a little diagram of SARS-CoV-2, the genome. Let's say everybody doing biosurveillance is targeting region A, and you target region B, then it's very short regions. There might not be full genomes available, so you won't be able to compare what you've detected to what other studies have detected. Specifically for coronaviruses, and you're all welcome to go read this review by Dr. Geldenhuys, looking at surveillance for coronaviruses in Africa. So a part of the review looked at how assays are selected for biosurveillance. And as you can see, a large number of them used actually existing assays. Some of them updated the assays, and there are a few that newly designed the assays. So the universal region here, that refers to here, is one that is targeted by many assays that are updated. So it might not be the exact same assay, but it targets the same region, and the results can be compared between the different studies. While people using non-universal regions have a problem with comparing what they have found. So the most frequently used assay for coronavirus biosurveillance has been variations on the assay published in Watanabe et al. And this has mainly been due to the PREDICT initiative with global testing for paramyxoviruses. But Dr. Gelderhuys, we are in-house. We are using an assay that was updated from the Wu et al. Paper, because it's important to also consider the fact that some of these assays were published, like for instance in 2010. Some of them were even in 2007, and people are currently still using it. So there's been a wide diversity of viruses that have been described. These assays are not updated, and you might be missing out on some of the diversity. More recently, there's been a new assay published by Holbrook et al. With a high sensitivity that can be used for coronavirus biosurveillance. So just to mention some of the other viruses, as Prof. Markotter mentioned yesterday, that are also zoonotically important viruses that we are specifically looking at in our research, which are the paramyxoviruses and the filoviruses. So just to give you a summary of some of the assays that have been generally used. So there's been a conventional RT-PCR that has been widely used for paramyxovirus surveillance, targeting the orthoparamyxovirinase subfamily. And then a few others that have sporadically been used, but this has allowed us to compare the different findings from different studies. And I included some of the references here for you. Then for the filoviruses, there are several assays available, not only conventional, but also real-time PCR, which are pro-based. None of them are actually pan-filovirus, and we are currently busy developing a pan-filovirus real-time PCR assay with collaborators at NICD for the detection of filoviruses that we will be using for biosurveillance. Some laboratory requirements. So when you enter into the lab and you want to start testing the samples, there are a few equipment and things that you will require in the laboratory. That is a biosafety cabinet or PCR cabinets where you can do your work, centrifuge, a pipette, a heating block, which are some of the things that we also require in the field, and then a thermal cycler, which can be a regular PCR machine or a real-time PCR machine. I will explain a bit about that later. A vortex, electrophoresis equipment, and then I say a sequencer or a sequencing provider, because sequencers are quite expensive and usually smaller labs don't have their own sequencers but outsource these things. But it's important to keep that in mind. So a lot of you might not be familiar with molecular testing and things like that. So I'm going to try and explain this so that everyone can understand. I won't go into much detail. So viral genetic material can be either RNA or DNA. The ones that we've been focused on, the coronas, philos, paramyxus, are RNA viruses. So regardless of what the genetic material is, you have to extract this from your samples that you collected. So there are different kits or approaches that you can use, which can be specific for your target. Let's say the RNA extraction for RNA viruses, DNA extraction for DNA viruses, and you also have kits that allow you to dual extract this. So for funding, how can I explain this? So you can actually save a lot of money by using dual extraction kits if you have the resources available, and have DNA and RNA for the samples. Because in future research, you might want to look at the presence of DNA viruses within the samples, and then you already have the material available. Especially for smaller bats, we do tend to have smaller sample sizes, so you can't necessarily go back and do another extraction of DNA from the sample, because you waste valuable sample. And then dual extraction kits actually work very nice to get both DNA and RNA out. It's also nice to have the DNA, because if you fail to do barcoding on the wing biopsies, or you didn't collect wing biopsies, you can use the DNA, then extract it from, for instance, a kidney sample that you're now testing for a specific virus, to actually do barcoding and identify the host species. So in this way, we can maximize some of the outputs at a lower cost. So specifically for RNA viruses, we do need to convert the RNA into complementary DNA to be able to do the PCRs and the assays on these samples. So this can be done in two ways. You can either make your cDNA to be specific for your target virus, or you can make it random. So specific would just mean that you would use primers specific for instance, coronaviruses, where random would mean it would take, you make cDNA from all the RNA within the sample. So if there's other viruses present, if there's anything else RNA present, you will be able to make cDNA from that. The reason why we try to use this is because cDNA step in testing is quite expensive. And by making random cDNA, we can actually use it for testing coronaviruses, paramyxoviruses, and filoviruses from a single cDNA reaction, as compared to doing it times 3. So that would roughly, in our currency, relate to 100 Rand compared to 300 Rand if you're testing the samples. Then for the assay, specifically PCR or qPCR, this will depend on your assay selection. And in many instances, the viruses are present in the samples at low viral copies. So that's why we tend to use 2 rounds of PCR to try and amplify the virus. And what I mean with PCR is the exponential amplification of your virus. So you would have specific viruses, primers for your viral sequences that would bind there, and you would amplify that region. And then the next round, it would be an exponential amplification all the way through to the end of your run. We do have limited sample material available, and we can't continue to repeat things, trying to see if it's positive or not, and that's why we do include a nested round of amplification, where we take the product from the first round and do it again, and we tend to get better results using that. So as I mentioned, you have 2 types of machines that you can use. The regular PCR machine that is usually used for regular first round and nested round PCRs. But for real-time PCRs, which is just another approach that is pro-based, you can use a real-time machine. The difference is the cost in the equipment. A real-time machine can be quite expensive, but you usually, it's a single round of amplification and not a nested round. So there you save costs on reagents, but the initial startup cost is quite expensive. Okay, so, and then agarose gel analysis. As I mentioned, you need a gel tank and a transilluminator to view these results. With real-time or qRT-PCR, you can actually see if your sample is positive or not. But with regular PCR, you will have to analyze the samples on agarose gel to be able to see was there amplification or wasn't there amplification. So I will discuss this part further from after you reach that point under the section for virus characterization later on in the talk. Then moving on to serological sample testing. So that would just plainly be collecting of your serum samples, looking at the presence or the absence of antibodies, and which will give you information on the immune status of the virus. So what I would, so the target of antibodies is usually an antigen, and the antigen would be, for instance, the surface proteins of your viruses, for instance, or for example, spike proteins in coronaviruses or glycoproteins in paramyxoviruses. So you get different approaches that you can use for serological analysis, which is your ELISA or enzyme-linked immunosorbent assay. So this allows for the detection of a specific protein or antibody against a specific protein. And amplification is usually by a signal of an enzymatic reaction. Then more recently, as also mentioned in one of the previous talks, more updated technology is the Luminex or the Bioplex technology. That is bead-based, where you can multiplex your assays to detect up to 100 antigens or analytes that you're looking at. However, if you consider the equipment required for an ELISA, you have your ELISA plate reader compared to the Luminex machine that actually runs the bead-based analysis. And these machines are quite costly. In South Africa, we're looking at upwards of 1.5 million just to get the machine. But in the long term, if you think about it, and you can do multiple analytes on small amounts of samples, you actually save a lot of cost and labor and time compared to running individual targets on your LISA. Considerations for this would be proper positive and negative control sera, and that is actually a problem for a lot of the bat work that we do, is actually finding or having positive and negative control sera specific for specific species and specific viruses. Also, considerations should be available antigens. We specifically are working on obtaining gene sequences for important antigenic regions like the spike proteins and the glycoproteins to be able to use this in designing these assays. But for a lot of the bat-related diversity of viruses, we still don't have these antigens available to develop these serological assays. Then just briefly into another approach that can be used is virus neutralization tests. For this, you require a virus isolate or a pseudovirus particle that contains the antigenic sites of the virus that you're looking at. It does require the propagation of the virus in cell culture, but with this assay, you will only be able to detect antibodies that's capable of blocking the replication of the virus, and not any of the other antibodies or other antibodies made for the virus. So you do rely on the presence or the absence of CPE, which is a cytopathic effect in your cell culture, like cell death, or the use of a dye or a stain to be able to see if there's viral replication. Or you can look at viral replication using RT-PCR to see if there is actual increase in viral titers or not. So considerations would be biosafety, especially if you work specifically with viral isolates of high biocontainment viruses, for example, Nipah virus that was mentioned yesterday. And requirements would be a biocontainment facility, obviously if you're working with high biocontainment viruses. Then cell culture incubators, biosafety cabinets, a different type of pipette than you would use for molecular work. And then you also have a lot of consumables and things that are a bit different. Then we move on to the identification of the virus, so initial identification. So you've now analyzed your samples in the lab. You've seen either on the real time or through agarose gel analysis that some of your samples came up positive. There is a chance that there might have been contamination with your controls in the samples. So how do we distinguish between that? Is we do sequencing. We actually sequence the DNA, the amplicons that we have, to see whether it is actually virus or whether it's contamination, and if it's virus, what is it similar to? So in general, for this you would use Sanger sequencing. As I mentioned, this is actually very specialized and expensive equipment, so it's not readily available in all the labs. And we make use of external parties where we send this to for sequencing to be done. So you would then receive back or a sequence, and you now want to check what it is. So there are a few online databases that are available out there, and I've listed here for you one that we use, which is GenBank database, where we use the BLAST function. So what you do is you would put in your sequence that you've now received from the sequencing facility, and you blast it against a range of sequences that are available in this public database, from different viruses that have been characterized, from ones that are detected through other studies. And then you can see, for example, okay, this is not very clear, but that says bat coronavirus, isolate this. You can actually go and see, oh, it's a sample that was tested from Madagascar, for instance. And then you can, okay, then you can see, okay, your sample is positive, and you can work further from there. Then at least you know it is a coronavirus, it is a paramyxovirus, and that just gives you some basic identification of what you actually amplified from your sample. So for bioinformatic analysis, as I mentioned earlier, your cross-sectional studies will be able to give you some information on the diversity of viruses. So with studies like that, you will be able to perform phylogenetic analysis, see the relation of your virus to other viruses that have previously been reported, and even look at putative species determination. Is the things that you detected novel? Is it similar to other things? And things like that. Then also, if you have data on longitudinal studies, you will be able to look at things like excretion dynamics, how different viruses are excreted differently over time, how are they excreted differently in different sample types, as well as considering the diversity that you have detected. So just some considerations is for things like data modeling, you do actually need some expertise. And in terms of software, a lot of the software that we use are actually freely available from the internet to use. You do have other programs or other online cloud services that can actually do some of the analysis for you. But I think it's nice to know that the software is available out there, it's free, anyone can use it. So just a little bit on additional characterization. So if we want to characterize a virus, we can look at obtaining full gene sequences, for instance, a polymerase or a spike protein, or we can look at complete genome sequencing. So there are a few approaches that can be used. So I just labeled the first one here as conventional PCR. That would be similar to the methods we previously described for you to use during testing of your samples. It's the similar techniques and equipment and things that you will need. So there can be short region amplification, where you can amplify regions of up to 1,000 base pairs, and you will have overlapping, or your assays will overlap to in the end give you a complete sequence. This can be used in combination with Sanger sequencing, but will require a lot of sample material, and we don't always have that available. It's more labor intensive, and it can actually be quite a problem if the target that you're looking for is a quite diverse virus, and we don't have anything to compare it with to design the assays with. Then another approach, also still part of your conventional PCR, would be a large amplicon sequencing, where you can actually design your primers to span larger regions of your genome. But if you do that, you won't be able to sequence it with Sanger sequencing, and you would have to look at another approach called next generation sequencing. This will require less sample material, obviously, as you have less assays that you will be running. But if your viral titers or your copy number in your samples are quite low, you tend to struggle with getting the larger regions. Or if your integrity of your sample is questionable, you will struggle to get this. And again, there is the problem of if your target is too diverse, what do you do? So the second one would be deep sequencing, or as I just mentioned, next generation sequencing. So if we, this approach is non-biased. It's not specific for a virus. If you take your sample, you send it in, it will amplify whatever is in the sample, and it will give you sequences for whatever is in the sample. And as you can imagine, with Sanger sequencing, you get a single sequence out. Compared to this, you can get millions of reads. So it can be very costly and very computationally challenging to be able to analyze the data from studies like this, and you do require specialized equipment. But there are facilities out there where you can send the samples to and get this. But if you're sitting with millions of sequence and you have host in there and whatever was in your sample, you will struggle to get viruses out, or viral sequences out. There are, however, methods to enrich for your virus, but you do stand the risk of losing some of your copy number of your virus because it's already in such a low concentration. Then just uses of what you can do if you further characterize your virus, which is like you can express antigens that can be used for serological assays, like if you have the sequence for the spike of the coronavirus, this can be used to express these proteins that can be used as antigens for serological assays. Characterization can be used to improve virus classification. We can do studies on viral fusion, attachment, and entry. We can look at recombination analysis, and you can also look at evolutionary analysis of your viruses. And that concludes the session. Thank you.